Endosperm. Endosperm is a product of post-fertilization. After the occurrence of double fertilization in sexually reproducing flowering plants, there is formation of zygote, which will be developing into embryo, and the developing embryo will be nourished by endosperm. Endosperm is formed from primary endosperm cell. As you can see here on the right corner of your screen, you're shown with cut section of seed of maize or seed of corn. Within the seed of maize, you are able to observe endosperm. Endo refers to inside, and sperm here means nutritious substance. So endosperm is nourishing developing embryo. Here in case of maize, we have embryo which is represented by plumule, which will be developing into shoot, and plumule is surrounded by a leaf-like structure called coleoptile, and we have radicle, which is the root, future root. Radicle will be developing into root system, and radicle is enclosed within a leaf-like structure called coleorhiza. And in between the embryo and endosperm, we have a region called scutellum, which is the cotyledon of maize. In case of monocot, we call scutellum for cotyledon. Usually in some seeds there is absence of endosperm, and those seeds which are having endosperm are known as endospermic seeds, and those seeds which are not having, are absent of endosperm, are known as non-endospermic seeds or non-albuminous seeds or X-albuminous seeds. We'll be discussing later. So bean seed is not having endosperm. Now endosperm usually develop earlier than embryo development, and as we discussed, primary endosperm cells will undergo repeated mitotic division to form triploid endosperm tissue, which will be acting as food reservoir for developing embryo. So endosperm act as reserve food material which nourishes the developing embryo. There are different ways of development of endosperm. Endosperm usually develops in three different manner. They can be nuclear endosperm development, or cellular endosperm development, or it can be halobile endosperm development. Regarding the free nuclear endosperm development, it is one of the most common endosperm development. As you can see here, in the free nuclear endosperm development, first there is occurrence of continuous karyokinesis. Karyo refers to nucleus, kinesis means division of nucleus. So there is continuous nuclear division occurs, and they form many nuclei within the single cell. And after division of nuclei, then there is formation of cell wall, or there is cytokinesis occurs. And in a way, there is formation of mass of endosperm, cellular mass of endosperm. So in free nuclear endosperm development, first there is occurrence of continuous nuclear division, and then there is cellularization occurs. There is cell wall formation occurs. Okay, and regarding cellular endospermic development, we have cells after each karyokinesis there is occurrence of cytokinesis. Regarding halobile endosperm formation, there is mixture of free nuclear endosperm development and cellular endosperm development. First there is occurrence of free nuclear endosperm development, and then there is cellularization occurring. So both the free nuclear and cellular endosperm development is found in halobile endosperm development. In case of coconut water that we drink from tender coconut is one of the best example for free nuclear endosperm development. The water that we drink from tender coconut is free nuclear endosperm. And after the cellularization, after the formation of cell wall or cytokinesis of these surrounding of the free nuclei, then there is formation of white kernel that we eat. So chewy white soft kernel that we eat from coconut is the cellular endosperm. As you can see here, in case of coconut, you are able to observe exocarp, the outermost covering of coconut. Then we have mesocarp, the middle covering, and we have endocarp, which is the innermost layer of covering. And within the endocarp, we are able to observe the solid endosperm, which is the cellularized endosperm. And within the solid endosperm, we are able to observe the liquid coconut water, and which is nothing, it is nuclei, free nuclei without cell wall, and you are also shown here with embryo. Now we have said that endosperm may be completely consumed by the developing embryo, and we may not find endosperm in mature seeds in case of some seeds, and those are say for example we have in pea, groundnut and beans, there is no endosperm. So the seeds of pea, groundnuts and beans can be called as non-endospermic or exalbuminous or non-albuminous seed. But in case of some seeds, say like coconut and castor, we have endosperm in the mature seeds. So endosperm may persist in mature seeds, and these are known as endospermic seeds or albuminous seeds, or we can say these as endospermic seeds. So this is all for your information regarding endosperm. We'll continue with development of embryo. Thank you. I'll see you in another video.